Welcome, everyone. I'm your host, Doug Erickson, Executive Director of Santa Cruz Works. We'll start in just a minute, but before we get started, I wanted to let you know you can post questions to the presenters in the Q&A. You can use the chat to chat amongst yourselves. Our event tonight, COVID-19 Mutations in You, is designed to answer your questions about the growing number of COVID-19 variants. Yes, variants. Very scary, I know. Variants have emerged in Britain, South Africa, Brazil, and now in California. The evidence so far indicates that they are much more infectious than the Italian strain, which has been racing across the globe since February of last year. Today, you may have watched President-elect Joe Biden hold a memorial service for the 401,000 US COVID victims. I quote Biden, "To heal, we must remember." Boy, do we have a lot of remembering and a long way to go before we heal in both body and mind, economy, social equality, and our education and political institutions. Biden has promised his administration will lead what I believe is the vaccine we so desperately need, science and truth. Here to bring us science and truth and to introduce our speakers is Isabel Bjork, the Executive Director of UCSC's Genomics Institute. Welcome, Isabel.
Hi, everybody, and thank you, Doug. Welcome to our panel, everybody. I'd like to start by just thanking Santa Cruz Works for putting this panel on. I have to say that I am constantly impressed at how Santa Cruz Works puts on panels with speed and extraordinary relevance about the issues that are dominating our thoughts in the immediate moment. And there is none that is more important right now, I think, than this one. And so specifically, looking at the virus that causes COVID-19, the virus mutations, and understanding how these affect all of us. So with that, I'd like to get started because we actually don't have very much time together tonight. And as noted, this is critically important to all of us. Just a couple process issues. Doug mentioned that there's a Q&A. When people registered, there was also an option to send questions. We've looked at those questions. We've incorporated them in the presentations tonight, but we will also incorporate them in separate questions if they're not covered by the panelists in their presentations. Know too that you can send questions throughout and we will look at them throughout. We're saving 20 to 25 minutes at the end for questions, but they may be directly relevant to what the panelists say. So I may interrupt the panelists to ask those questions. So with that, let's get on to our distinguished group of panelists. And the panel's made up of medical professionals and scientists who are particularly familiar both with the virus and the concerns around the impact on the community here in Santa Cruz and the international and national impact. So first, we have Dr. David Ghilarducci. And David is one of the key actors in the Santa Cruz County Public Health Department's COVID-19 response. We're very lucky to have him here with us tonight. He's also the Emergency Medical Services Director for the county. I will add that in 2010, David worked with the International Medical Relief Corps helping in Haiti after the earthquake. And I mention that not only because it's near and dear to my heart, because I have a background myself in humanitarian aid, but also because that background and looking at what's happening internationally and the impact of people is so important with respect to COVID-19 in particular. We also have here Dr. Amy McEntee. She's the Chief Medical Officer at Salud para la Gente. Salud is a community clinic that covers Santa Cruz County and North Monterey County, and it has a particularly strong presence in Watsonville, where, as I think most people know, some of the highest numbers of COVID cases in the Santa Cruz area have been experienced. We also have Professor Beth Shapiro. She's a professor at UCSC in ecology and evolutionary biology, and she specializes in conservation genomics and paleogenomics and is an expert in sequencing. And we have here David Hausler, who is also a professor at UCSC. And he is not only in biomolecular engineering, but he is also scientific director of the Genomics Institute at UCSC. And both Beth and David are Howard Hughes Medical Institute investigators, which is a very particular honor afforded a very few amount of scientists in this nation. So with that, I would like to start with David Hausler. Welcome, David.
Thanks so much, Isabel. It's great to be able to talk to everybody about this important issue. And so I think Doug gave us a great introduction. One of the things I will say is what you hit right on the head, Doug, was the scientific approach. And that's what we'll be talking about tonight. We're really taking a scientific approach to this, and we'll describe to you how that works. But the other thing I want to say up front is don't panic. So this is not a time to panic. We don't know the severity of these variants, but it seems very unlikely that any of the variants that are there right now will, for example, make the vaccines not work. So it's not a time to panic. The variants, though, as Doug said, are now proven, some of them, to make the virus spread easier. And some are speculating that that may be an explanation for the recent surge in the virus. So when we talk about a variant, I want to talk about three things starting with it. First, a mutation. So the virus is a bit of RNA. RNA is the cousin of DNA, ribonucleic acid wrapped up in protein. And the RNA is the message it carries into the cell to make more copies of itself. And that message can mutate. So we know about DNA mutations. RNA has even more mutations. And in fact, viruses evolve that way by accumulating mutations in their RNA message. Now, one of the great things about what's happening now is that we can read that RNA message. It's about 30,000 letters long, and every virus carries one. Everyone who gets infected has a message written in RNA in their virus particles, and we can read it. And I am incredibly excited about this capability because this is really the first time we've been able to do that at a huge global scale. I remember back at the 2003 SARS outbreak, we were lucky to read a dozen different virus genome sequences from different infected people and try to get an idea of how this message varies from infection to infection. Now we have over 200,000 different virus sequences that we can access in globally shared databases. And that allows us to watch the virus in detail as it changes, as it evolves. So I have a movie to show you, to give you the actual experience of watching the virus. Now, so can I share screen? I'm doing that.
Yes.
And here we go. This is a map of the world. Each of these circles represents the COVID cases that are in that region. Usually it's essentially a whole continent or a whole area. And the pie chart shows the different types of mutations that are present. So that can tell you that not all viruses are identical and they change through time. Now, this movie was made by this wonderful group called NextStrain that works with us, and we've been on at UCSC since February 7, tracking these things. If you go to our UCSC SARS-CoV-2 genome browser, you can track that since last February 7th. But this movie was made by our friends at NextStrain. Now watch, starting from 6 months ago to end of December, this is what happens. Through time, you see as the colors change in the pie chart that some variants become more common or not. But then this orange variant comes up, B1.1.7, and look at how fast it spreads. One more time. So this gives you a feeling, and it's the feeling we all went through. We watched for months and months as the virus mutated very slowly, on average about 1 new mutation per case. And none of those became dominant very rapidly until this orange variant, B.1.7, showed up. And then bam, it spread much faster. So over a period of a month, you see a very rapid spread. That is the indication that this orange variant actually contained changes that made the virus spread more rapidly. Now we can actually watch these things, and this is what tonight is about. You have probably been reading the newspaper, and as Doug said, we have a new one coming up from South Africa, from Nigeria, from Japan, and so forth. Even in the United States, there was a big report of one that started possibly in Houston a few months ago and is now becoming very common in the US. So what has happened in the last few months is that the virus has started to accumulate sets of mutations. And a set of mutation defines a variant. So if a virus accumulates lots of mutations at once, we call that new mutated version a variant. And if that variant causes the virus to spread faster, it can spread quite dramatically. But the other thing we're concerned about is shown in the second figure here. Okay, here it is. This is the SARS-CoV-2 virus. The RNA message is inside this sphere, and on the outside, these little orange mushroomy things are what's called the spike protein. This spike protein, if you magnify it, is the actual piece of the virus that recognizes something on the surface of a cell. So this is a cell, maybe in the epithelial lining of your lung. So somewhere in your lung, there's a cell and it's displaying this blue thing called ACE2. And this virus has evolved to stick to that. It sticks like glue, just a perfect fit. And that is what pulls the virus into the cell and sets off the infection. What happens when you get vaccinated is that you make antibodies, and antibodies get in between in this pink area and they gum up the works. They prevent the virus from infecting your cell. So this is great. If you get infected, you will naturally make antibodies. But if you don't make enough or the right kind, you will get very sick and need some help. You might need to go to the hospital. In the hospital, they may treat you with antibodies that come from other people or are manufactured, and they may treat you with other therapies that will help you overcome the virus. The problem that we're worried about now is that if the virus changes, the antibodies which previously blocked it from contacting and getting in the cell will no longer recognize and block it. It's as if the virus changes its coat and now the immune system can't see it anymore. This happens routinely with influenza. So influenza is also an RNA virus and it changes more rapidly. And we have to get a new vaccination every year because the influenza virus has changed its coat in some way. And our antibodies, which were great for last year's strain, don't work so well for this year's strain. We're not sure that SARS is going to be that way. In other words, every year an update, But now with the rapid emergence of new variants, we are sure that this RNA virus can change. And in particular, we see changes right in this part of the protein, which is the business end of the protein that is key to getting in our cell. So we have seen in 3 separate instances now the same change that made the virus a little more evasive to existing antibodies. And we have many, many tests on the genome browser to show this. So we will see something like the flu, and we're here to tell you, don't panic, just like we handled the flu, we can keep track of these things, and we will keep track of these things. We are starting to begin to sequence all of the variants of the virus, including the variants that are common in Santa Cruz, and we will be able to recognize when dangerous changes to the virus happen, if indeed they do happen. And again, let me close by saying, up until now, the only changes we've seen are ones that we're sure are making it easier to spread the virus, but not making you sicker when you get the virus, and not preventing the vaccine from working. But we're on guard for looking at those other kinds of changes. All right, so that's the introduction and a little bit of the science. I hope you enjoyed the pictures and the movie. I will let my colleagues finish off this evening.
Thank you, David, for that background, which sets us up perfectly for the next panelist, which is Dr. McEntee from Salud para la Gente, to talk about how this information about new variants of the virus and questions around transmission have affected her medical practice at Salud, her concern about the patients and the population that Salud serves. Welcome, Amy.
Thank you. Thank you for the introduction. I'm Chief Medical Officer at Salud para la Gente in Watsonville, and we started testing for COVID in March, and then we're able to increase our testing quite a bit once we partnered with UC Santa Cruz. At this time, we're testing about 100, sometimes more patients per day, and we've consistently had a high positivity rate for the county. With the first surge in July, we were testing around 30% positivity rate, and now we're at about 40% positivity rate. When we learned about, part of our role with the testing as well is we have a contract with the county to help with contact tracing and investigation. When we first learned about sequencing from UC Santa Cruz, we were most interested in seeing if it could help us better understand how we're seeing the virus move through the community, and if the preventative measures that we're recommending are working, and if we could better understand that, if we're able to better prevent spread. Additionally, when we've had some cases where it's hard to figure out, it seems that everything was done correctly, and how did we still see this spread? Or are we seeing, when we're looking at a cluster, are we looking at more than one cluster, or is it actually spreading from a couple of different directions? So really looking to see if this type of information can help us figure some of those things out.
Thank you very much. One question that came in was just asking what you would do in particular in terms of the information around sequencing. So David, for instance, was talking about how we are starting sequencing. Are there thoughts with Salud around how to address that?
On a like individual patient basis, I think, is that?
Or a policy basis, either one.
I think probably the policy basis is more related to public health, with recommendations on social distancing and masking and hand washing, and if we're able to see if things are moving differently, if that impacts what we do and what we advise people on. For us, seeing this would be most helpful is we have had an instance where we had a fairly large cluster where when we're interviewing people, everybody's, they distanced, they wore a mask, they hand washed, they followed all of the directions, yet we still saw this spread to several people. And we're not understanding is, are we not getting some of the index cases? Then maybe this was more than just one person that spread it in a workplace, or was this spread rapidly for another reason? So that's really where we're looking to see if that would help. The other question that we have is that it's rare, but occasionally there are reinfections. And if we see somebody who had COVID maybe 4 or 5 months ago, and then they're re-exposed and we're testing them again, could this help us inform us if this is a new infection or if we're still seeing a positive from the old infection?
Thank you very much. All right, so let's move on to our next panelist. And that goes directly to what Dr. McEntee was talking about in terms of the policy making side of things. So Dr. Ghilarducci, this is time for you to present on your role in public health, and particularly what the major concerns are in that role and what you're seeing with regard to the virus and the variants that are being discussed.
Well, good evening, and thank you, Isabel. Well, first, I needed a full disclosure. I'm a graduate of UC Santa Cruz back from the 80s, and I've just been in awe with the response that UCSC has had in terms of helping us with this particular pandemic. And I remember back working in a plant physiology laboratory in Tiemann, and next door was Harry Knoller's lab. And for those of you who know Harry Knoller, was a real pioneer in ribosome research and RNA research, which has really, I think, led to the development of the kind of vaccines that we have today. So, this really started years ago when Chancellor Sinsheimer called for the sequencing of the human genome is an incredible task that I think really started here in Santa Cruz. So just a testament to that. Also, I want to just shout out to Salud para la Gente, has been a fantastic partner with public health, especially with respect to the equity lens that we all need to keep in place. In fact, this has been a pandemic that has disproportionately affected our communities of color and have exposed, I think, some of the underlying sort of disadvantages or flaws of having a patchwork medical system that we have in this country, as well as the underfunding of public health over the years. So where are we today with the pandemic? It hasn't been worse than it is now. And we're seeing signs that it's going to continue to get worse. We've been very concerned about a post-Christmas and New Year's surge. We're hoping that that doesn't come to pass. But right now our hospitals are full. I check with the hospitals every day. And on any given day, our ICU capacity is 0. Or sometimes we have patients in line waiting to get into the ICU. So that's a remarkable state of affairs. And Isabel, you mentioned Haiti, and it reminds me in some ways of that. I know that sounds like an extreme thing, but it reminds me of being in a situation where we had to ration care because there simply wasn't care available. And here we are in the United States where ambulances are being told to conserve their oxygen. Hospitals have had to shut down for a while because their oxygen systems froze up. So this is unprecedented for the state of California and for the United States. Just to put it in numbers, if you remember back when we were in purple tier, seems like a year ago, but it was a couple of months ago when we reached the purple tier. The threshold for that was 7 cases per 100,000. Today we're at 70. So we're an order of magnitude higher in terms of rate of spread. And our wastewater data is showing that this is continuing. So if this is due to a variant, I think that would be very interesting to us from a policy perspective, because we need to understand what conditions are leading to spread. And maybe we need to adjust the advice that we've been giving. Maybe 6 feet is no longer enough. Maybe it needs to be 10 feet. Maybe we need to consider other kinds of controls, like instead of a cloth mask, we use a medical mask. Lots of open questions. And we've been in conversation with epidemiologists at the state level, who, by the way, predict that the B117 variant will probably be widespread throughout California by mid-March. So it'll be very interesting to us to know if that variant is here, and then how that might affect policy from the public health perspective. If it is circulating in our communities of color, then we would expect that people who, anywhere really in the community who are maybe able to get away with a basketball game and not catch a virus, or they were able to have a backyard barbecue and be just fine. This variant may actually change that equation and make it much more likely. On top of that, of course, there's just a higher prevalence of virus floating around in the atmosphere in our county than more so than there ever has been before. So this work is really going to be fundamental into understanding the policies that may need to be adjusted as we get through this and until vaccinations become widespread through this country. This is kind of a crisis that we need to address.
Thank you very much. So to build on that, in order to think about the policy further in terms of analyzing what the variants are telling you and all of us, we need to be able to get that information. And so our next panelist is Professor Shapiro, who can talk about how the sequencing is done and give us some background on what it means to do virus sequencing and the information that it can provide. Welcome, Beth.
Hi, thank you. So we've heard David Hausler talk a bit about the new variants that are emerging, and then Dr. McEntee and Dr. Ghilarducci talk about how understanding where these variants are, how prevalent they are in our community could be useful in designing or updating policies, or just really better understanding how what we're doing right now is working to control the spread of the virus. I think the question that comes up about the sequencing most commonly, because we hear about this and we think, well, this seems like a no-brainer. Of course we should be doing sequencing, but why haven't we been doing it? Is this something new? And the answer to that is that it's not new. It's not new to science to think about sequencing genomes, and it's not new to think about sequencing genomes to try to answer these specific questions. I've been working in paleogenomics for a long time. We extract DNA from lots of things, including things that have been dead for a really long time, like mammoths and giant bears and stuff like that. But viruses have, for a long time, been a source of information about this. We understand about the origin and spread of things like HIV and influenza. Even the last pandemic, the 1918 flu, we understand where it came from and how it spread because we generated genomic sequencing data from these viruses, and we're able to then line up all of the sequences that we had collected from across the world and figure out how they were all related to each other, and then use the tools of population genetics and things like that to figure out what this means. And this is what we would like to do, what we need to do with this virus. It's not something new that we need to worry about, being different or somehow dangerous. We know how to do this. So why haven't we been doing it? One of the reasons that we haven't really implemented it yet in the States, Dr. Ghilarducci talked about how we obviously have a vastly underfunded public health system, so that's one thing. We also care very deeply about privacy, and we are worried that because we care so deeply about privacy, we might be getting information that we don't want to get out if we're sequencing DNA. And this is, I think, the most important point that I need to make about this. When we're sequencing these genomes, these viruses, we are only sequencing the genomes of the viruses. We don't capture any human DNA. We cannot say with the data that we get what human the sequence came from, but we can say that this sequence came from perhaps this zip code. That's something that we would like to say, and was isolated on this date is also something that we would like to say. The way that this works, we get the positive test, and that's an RNA sample. David explained that the virus is about 30,000 letters of RNA. And then the sequencing it uses the same sort of approach. The positive test is PCR. It's an amplification. And the reason that we use PCR is because it is very sensitive, just a very tiny number. Theoretically, one virus is enough to be able to get a positive control. And it's also sensitive. It won't pick up any other coronaviruses or any other viruses that are infected. So it's sensitive and also specific. But the sequencing then will take that sample that we know is a coronavirus sample, take that long virus and chop it up into little fragments and sequence all of those and then piece them back together and just allow us to say, okay, here is the 30,000 letter sequence that infected a person from this zip code on this date. And then we can collect all of that information and we can line them up on a computer and we can ask where they're similar and where they're different. And because we know that only a small number of mutations happen every time the virus goes from one person to another, we can use that information to reconstruct where we see clusters or perhaps where the origin of different strains are. And we certainly can identify some of these new mutations and identify or new variants that are circulating, not just the B117 strain, but also the strains from South Africa, et cetera. There are new variants popping up and we would like to know where they are and whether they're spreading. So this is the type of thing that we can do by sequencing that we can't do by just getting the positive PCR. And this is what we are working to provide to Salud and also to the community.
Thank you, Beth. I'd like to start the question and answer period now because we have quite a few questions that have come in, and I thought it would be useful based on what you were saying, Beth, just to start the first one with you. So you and David Hausler touched on this already, but explicitly, maybe you could explain this. So are you surprised to see variants emerging after a year of the virus existing, or did you expect to see variants earlier?
Well, we have seen variants earlier. The video that David showed that there are lots of variants that are emerging all of the time, and these variants will drift to higher and lower frequency depending on whether they make the virus more transmissible. Just in evolutionary terms, the virus is more fit, more able to reproduce and make copies of itself. Most variants, just like mutations that arise in our own genomes, we all have 40 mutations or so that are different, that arose just in us, that neither of our parents have. Most of those mutations don't do anything. And so it took some time for us to start seeing mutations that did change the transmission dynamics of these viruses. One thing that's interesting about these new variant strains is that they all seem to have multiple mutations happening at the same time, which has led some people to suggest, and we don't know, but it's led some people to suggest that this might be because you had a chronic infection where all of these mutations are happening in a single person, and the virus is in them, just replicating, really doing whatever it can to escape control. And because there's so many viruses, that from an evolutionary perspective, it makes it likely that you see these mutations. Another problem that we have is that we haven't, as a global society, gotten this virus under control. And this is bad also from an evolutionary perspective, because if you have a really big population size, then natural selection, this force that is trying to make the virus better able to be a virus, is a really powerful evolutionary force. And it's another reason to get the numbers of viruses low, to really get this under control as a global community. So we can reduce the possibility of seeing these more infectious variants. David probably has something he can add to that.
Go to it, David.
No, okay. He says no.
We can't hear you, David. To just the general question, there are a lot of questions that have come in the chat.
Sorry, that was brilliantly explained, Beth. Nothing to
There are a number of questions that have come in about what the problems that are potentially caused by the new variants, and particularly about what it means to be more transmissible. Beth, you've already touched on that a little bit. David, did you have anything to add to that?
So if you remember the picture I showed, the key part of infection is the virus getting into your cell. And that's what the antibodies prevent from happening. So you may be surprised, but it takes thousands of virus particles to have enough chance to even get one infection that will take off in your body. So it's the efficiency of how the virus gets into the cells in your body that is the key thing about transmissibility. If it only takes a tiny bit of the virus to infect you, then it's highly transmissible. If it takes a ton, like if somebody has to cough right the hell in your face for you to get it, then it's not so transmissible. So the key, we think, and again, let me put it this way, there's no hard evidence that it's anything but the virus being more effective at getting into your cells that make these new transmissible, more highly transmissible variants more transmissible. Now, there were some good questions about, would it be more transmissible by surface exposure or by air and so forth? People are working very, very hard to do those studies, but we can't tell from reading the genome whether, oh, this is going to be much more transmissible by touch, and this variant is going to be much more transmissible in a sneeze. We have to do empirical tests to determine that. It's possible to have mutations in an RNA virus that cause it to survive on surfaces longer. That usually requires a fancier set of mutations than just getting in the cell easier. And so we don't have evidence that it's somehow mutated to survive on surfaces longer, and therefore we have a new threat of type of transmission. There is no hard evidence about this yet. But again, we have to test these things.
Thank you. We have a number of questions that have come in on vaccines, and I would like to direct the first one to Dr. Ghilarducci. So we have questions, one question that came in is, what are the specific mechanisms of a vaccine and how do they operate in our systems? Would you be able to start with that? And then there's some follow-up questions as well for you and Dr. McEntee.
Yeah, happy to do a quick primer on immunology and vaccines. But essentially, what vaccines do is they intend to mimic the real disease in order to prime your immune system to have antibodies, or at least a memory of antibodies available, to be able to quickly respond to the infection or an exposure and prevent widespread infection. So usually it's a lot about antibody response. And as David talked about before, we actually can use convalescent plasma from people who have recovered from COVID and give those antibodies to somebody and help them recover, sort of artificially or exogenously apply antibodies for that purpose. What's different about the two COVID vaccines that have now been approved is that these are mRNA vaccines. And unlike some of you may remember or read about the Sabin and Salk vaccines for polio, one of which used a live attenuated virus and the other which used a killed virus. The mRNA vaccine does not use any virus whatsoever. There's no dead virus or attenuated virus. This is just simply a vaccine that contains the code of, or a signal for RNA, the signal for the spike protein, that protein that attaches to the ACE2 receptor that David showed in the diagram earlier. And so by just coding for that piece of mRNA, these vaccines actually use the machinery of the cell to produce more of these spike proteins, and only the spike protein, not the part of the virus that's dangerous or infectious. And then those spike proteins then stimulate the immune system. And unlike, say a flu vaccine or many other vaccines, what's really interesting and exciting about mRNA vaccines is they don't just produce antibodies, but they actually recruit other elements of the immune system, like the cytotoxic T cells, killer T cells, that actually I sort of think of antibodies as kind of a missile defense system, and the T cells are actually going at the missile factory, and so it really gives you a much better, more robust line of defense. And so I think that's why we're seeing 94-95% prevention of severe disease in these clinical trials, these phase 3 trials with these particular vaccines. So it's an interesting technology that's been around for a couple of decades, but this is the first time it's been applied to a vaccine in this way.
I'd like to add something to that too, which is another bit of good news, because I like to emphasize the good news here. If we do get a new strain, that one, let's say the Moderna RNA vaccine doesn't work for the new strain, then Moderna estimates that within a very short time, they could produce a vaccine for the new strain. I've heard as little as 6 weeks, which is incredible, incredible. We could never get a new vaccine that fast in the old days. So this is really a breakthrough, and you ought to be all really, really happy that we have these new vaccines.
Yeah, for example, the flu vaccine, usually the CDC has to decide in the summertime which strain of flu is going to be the strain of flu that hits the United States in the winter. And because it takes 6 months of using chicken eggs and so forth to develop the vaccine, this really bypasses that slow process and allows us to be more nimble.
Thank you. So, Dr. McEntee, there's a question here that sort of has two parts. So, the first part is just, can a vaccinated person affect a household partner that is sensitive? It says here to vaccines, but I think part of the idea too is, if you're vaccinated, can you also be contagious? And then sort of the next piece of that question is, as a medical professional, what are the discussions happening around what advice you would provide to people about steps that need to be taken post-vaccination?
So the first one, it sounds like, is if a person who is vaccinated, can they still spread this to other people? And I'll let Dave Ghilarducci add anything if he has anything to add on it. But my understanding is that once you've been vaccinated, it's unknown if you could still asymptomatically spread this to others. So even once we're vaccinated, we still need to continue to follow all of the safety measures that we're doing now, which is the social distancing, masking, washing hands. The studies for the vaccine really focused on prevention of serious illness, which they're very effective at that. And I would imagine as we have more people vaccinated and more information on this, things might change. But right now our understanding is that you could still potentially spread it, and you do need to take all the same precautions that you would take now. And then in terms of advice, like advice for patients, for if we're telling people to get vaccinated?
Both of those things. I mean, really, my question was more around what steps and protections needed to be taken once you were vaccinated, because we have several questions from the audience here about, transmissibility post-vaccination. What's the effect of vaccination?
So once people are vaccinated, we're still encouraging them to continue to take all the safety measures that you take now, because we don't know if you could be an asymptomatic spreader. One of the other things is that these vaccines are very effective, and they do trigger a strong immune response. So people do tend to have more side effects that are kind of flu-like symptoms that are not that you're sick, it's your immune system working. But with the prevalence of COVID, and especially with us being in phase 1A, where it's healthcare workers who are likely exposed more, it's hard to know if your symptoms are side effects or are these that you were exposed to COVID and maybe you have COVID. So we do, our staff that we vaccinated last week, we encouraged everybody that if you're having a fever and chills, like, we want you to come in and be tested, and we want to, find out, is this just a side effect from the vaccine, or have you been exposed? The vaccine takes, about 10 days to 2 weeks after your second one to be really effective. So there is the potential that you might have been exposed and you didn't know, and you could come up positive before the vaccines had time to take effect. And you could get COVID in between the two vaccines too. So it's important to take all the same precautions that we're advising people to take now.
Yeah, Amy said it perfectly. There is a misconception, I think, out there that the vaccine itself might make you infectious, and it's I just want to say it's biologically impossible to be infected by the vaccine itself. And so it is possible that you may have unknowingly had COVID when you got vaccinated, or maybe contracted it soon thereafter. That is not related to the vaccine itself. It's more of a timing issue.
Thank you. We have some questions here too around sequencing information about the variants, and why in particular it's taken so long to get information out there. And information out there means in national databases, for instance. And David, I know that you referred early on in your presentation to contributing information to national databases. Can you tell us a little bit about what that process is? You also referred to the UCSC genome browser, and maybe you could describe what that is and what it could do in this case. We can't hear you.
Dave, you're on mute.
Okay, so the world has really been on top of this virus in terms of sequencing much more than previously, but it varies from country to country. So like in England, they had a very, very strong program, well-funded program from the beginning. And in our country, not so much, really didn't spend any money on sequencing to speak of. So we have the capacity, it's a matter of the will to get the money out there and get the sequencing done. But despite the spotty kind of response around the world to sequencing, we have, as I said, built up a library of more than 200,000 different sequences, which is enough sampling of the virus to make that nice movie and many other movies that explicitly show the spread of the virus and show new variants as they come about. And we at UCSC took our human genome browser, which is used by about 10,000 medical and research professionals every day around the world for a wide array of human diseases. And we specialized it into the SARS-CoV-2 browser, which was first released in February 7th. We're coming up with the 1-year anniversary of that browser now. And that browser collected all the information about the molecular nature of the virus, including antibodies and ways that the virus was detected, and many other issues. For example, if there's a mutation in a place that's used to detect the presence or absence of the virus by PCR, as Beth mentioned, then the PCR won't work in the mutant version of the virus. And we keep track of that. We keep track of the antibodies and how well they bind. We even keep track of the killer T cell epitopes that are thought to be important in the virus. So a lot of molecular information was pouring in. And in fact, scientists are publishing papers faster than has ever happened before for any disease. It really has been a flood of scientific information. I've seen a chart that compares the growth in scientific papers to the growth of number of cases of the virus, and they can certainly be plotted on the same plot. They both kind of exponentially took off. But of course, there's a problem when you have too much information coming in, not all of it completely reliable. So we've worked with hundreds of scientists all around the world to filter through that and just put the information on the genome browser that is most relevant, most important, and most validated. So that information is used now by about 4,000 different scientists a month, more than 500 every day, and new ones showing up all the time. So one of the things that Santa Cruz has done is always argued for open, quick, rapid release of reliable information. And we've been famous worldwide for that for decades, since we posted the first draft of the human genome on the internet 20 years ago. So that's something I feel very strongly about. Now, there are some groups that want to restrict access to this information, and we've been arguing with them over that. You know, there has to be a commitment that we all work together and not try to control this information for our own gain, or for monetary gains, or for ego, like I want to publish first, so I'm going to withhold the information from others. These kinds of things are not good behavior in the time of a pandemic. And at Santa Cruz, we call that out, and we try to point out that it's important to share without restrictions, without use restrictions, without intellectual property claims, anything like that. You need to share all the scientific molecular information that's available with everybody immediately.
Thanks. So there are a couple questions about data and how much data is needed to understand the efficacy of the vaccines. And I'm going to throw this out actually to the panel because I'm not sure who is going to have the most information, or you may all, but specifically here's one of the questions, but they're all quite similar. So how long do you estimate it will take to have enough data on the newly found spike proteins to be confident enough that the currently used vaccines are effective against them? And the similar questions are essentially, how much data do you need to gather and how much time will it take before we have confidence in the vaccines that have already been rolled out?
David, you want to handle that?
Yikes. It really depends. I think, David, as you had shown in your presentation, it really depends on the configuration of the spike protein and whether the vaccine is creating an antigen that is similar enough to the spike protein in the variant that will continue to be effective as a vaccine. I can't answer how long it will take to know that. I think it would be relatively simple to do some tests, or at least some observational studies on once our vaccinated population goes up and we understand where this variant is, then we'll get a better sense of how effective it is. And I want to go back to another point. I think somebody had asked earlier, I saw in the chat, that wouldn't mass vaccinating actually select for a more pathogenic or transmissible virus? And I think what we're dealing with right now is that essentially the more virus that's floating around out there, the more opportunity there is for mutation. And so we really need to get a handle on this disease. We need to get a handle on the spread. We think that actually some of these variants are probably coming from people who are infected for a long time, perhaps an immunocompromised host, where the virus has a lot of chances to try different combinations to maybe escape immunity. So the fewer infected people we have, the less virus that is circulating, the fewer variants that we would expect. So vaccination is going to be a very important part of this.
I will add a little bit, and Beth can chime in here. It's important to understand evolutionary logic, right? So the virus is taking advantage of opportunities by this process of natural selection, and whatever works best for it gets more common because it works well for it and it gets it spread around. But to anthropomorphize this a little bit, the virus has no interest in killing you. In fact, quite the opposite. It would rather keep you alive and have you spread it to more people, quote unquote, right? I'm just saying it is more evolutionarily successful if it actually has a mild infection that transmits to more people. And so we often see that viruses do evolve to be milder, but more transmissible. So it's not, everybody has to understand the virus is not out to get you in some maniacal way, just to kill you for the hell of it, right? So given that that's true, we can make appropriate rational scientific decisions about what to look for. And one of those was to look for variants that were more transmissible and check to see if they're more lethal or not. But we'd expect not. And in fact, if you look at all the records from hospitals who've gotten data from people who have the new B1.1.17, they're no sicker than anybody else. So it's exactly following the logic we would expect. And we will, if we sequence people and we sequence more vaccinated people, we'll be able to see whether it makes a difference, whether you have B1.1.7 or you don't, or whether you have the South African variant or you don't. You just have to sequence and vaccinate and count.
Yeah, I'll just add to that, we don't only have sequencing and vaccinating and counting at our ready as a way of thinking about what's going on with these new variants. Over the last year, many of the labs across the world that have been focused on lots of different viruses have turned all of their knowledge toward trying to find a solution to this. And we have very sophisticated models where we can actually create in a lab the way that these proteins are going to fold on top of each other and look and actually see what the impact of these mutations is going to be on the virus. So we don't just have to wait and see. We can also do experiments in the lab that will tell us, even before people are infected, what we think is going to happen with the vaccines as we have them right now in these new variants. And that work is obviously ongoing.
Thanks. We have another strain of questions actually around serum antibody testing. And I'm wondering, and this one is actually directly to providers. And so Dr. McEntee, I'm wondering if you can take this one. The question is, what do providers tell patients asking for serum antibody testing? Specifically referencing what would you tell them post-vaccination, but more broadly, where does serum antibody testing fit into the considerations around the virus and patient care?
Sure, I can try. And if Dave has anything to add, he can add that. We really haven't recommended serum antibody testing. In terms of understanding what the significance is, I don't think there's enough information on that. We haven't recommended it, and we haven't recommended it post-vaccine. I don't think we know really what it means. I know that there is a lot of people out there that want to do that. I don't think it's really helpful clinically in terms of what you're going to do, or we don't really know what to make of the information just yet.
Yeah, I totally agree. It really has not been useful from a kind of contact or case investigation perspective. In fact, serologic testing can actually be harmful because it does take some time for the serology to show up the signs of infection. And so you could get a negative test on a serology, assume that you've never been exposed to COVID, when in fact you're just in the early phases, the early asymptomatic phases. So it would lead to a false sense of security. So we really haven't found serologic testing to be very useful, at least not clinically.
Thank you. And so going back to the graph that you showed, David, at the beginning, there's a question about the timing of when we'll have that kind of information that can be represented locally and regionally. So here we go. When will you be able to show infection spread locally and regionally like you showed globally?
Well, we want to get the information from Santa Cruz County as soon as possible, but that will take a few weeks now that the decision has been made to launch a sequencing effort, and that it is now underway. The results will appear only after a few weeks. And Beth, having been the one that's been key with her colleagues, Jeremy Sanford, Michael Stone, Mark Akeson, Miten Jain, Ed Green. I have to say, there's many, many names. There are many, many heroes at Santa Cruz. Russ Corbett. They've all jumped on board to make this happen and happen as quickly as possible. But of course, we can't just generate sequences overnight. But after the sequences are there, we will be able to get a picture of what's happening in Santa Cruz County, and we will be able to compare it to what's happening in other counties. Like Santa Clara is ahead of us, for example. They've been sequencing for quite a while, and they have a very good picture of what the variants are in Santa Clara County. We will catch up to them as soon as we can.
Thank you. I'm conscious that it's 8 o'clock. I wanted to ask the panel if there are additional points that you would like to make before we close.
I wanted to make just a quick point about vaccination and some of the things that we're encountering, kind of vaccine hesitancy and concerns about vaccination. I think some of the questions tonight were sort of along those lines of what is the risk of vaccination. And I think we're in a situation right now that the risk of contracting COVID and having a serious illness far outweighs any small risk associated with the virus. You may have seen some news recently about a certain lot of Moderna vaccine that seems to be causing more allergic reactions. We see the rate of allergic reactions to be really quite low with both of these vaccines, both the Pfizer and the Moderna, about 1 in 100,000. It is a little bit higher than what we're seeing in other vaccines, which is closer to probably 1 in a million. But that said, an allergic reaction is simple to treat. We do have medical teams at the vaccination clinics that are prepared to deal with that. It's reversible, causes no long-term harm, and you get the benefit of some immunity from the virus, which I think is super important at this time. So just wanted to reassure people in that way. There's also lots of myths out there about altering DNA or about being somehow derived from stem cells, or that you can catch COVID or test positive for COVID after the vaccine, and all of those are not true. So I just wanted to make sure that the public was reassured about that.
Thank you, David. There's no end of conspiracy theory out there. Don't believe it.
Yeah.
I'd also like to just say, as David started in the beginning, you know, we have reason to be hopeful. And this new approach that we're using, we really are going to dig into the sequencing. We're going to be returning these data to Salud and to the university and hopefully to other places in the county. And this will be useful. We will learn more about how the virus is spreading, and we will be better able to control it. And we will get our kids back to school, which, you know, I don't know if you've heard my running around in the background, but it's a really important thing. Like, we need to, we want to help. And this is an opportunity, and a great one. And we appreciate it.
And I want to add that you can help too if you're listening. Some members of our community have already stepped up with donations for this project. And if you would like to, it would be greatly appreciated. This is something that it's not in the budget for the county or the state or the federal budget at this point to help the county of Santa Cruz sequence. And the only way we can do this is if we resolve to do it with our own funds and our own blood, sweat, and tears. And I can tell you, people are stepping up like crazy to help with this project. So if you can help, if you really want to help, make a small donation to this.
Thank you, David, for that. And I have to say, one of the reasons that the people that are here on this panel are here is because every single one of the panelists here has stepped up and has been working incredibly hard. We talk to Dr. McEntee often, several times a week sometimes, about all the assistance that is being provided to the community here in Santa Cruz. And the dedication to address the virus is really quite incredible here. We had Dr. McEntee, I saw you moving for a second. Did you have a last comment? And then I will make a last comment too, but I don't want to interrupt.
So I'll just comment that, the partnership with Salud and with UC Santa Cruz and the public health has just been really impactful in the ability to respond to this pandemic. And we really appreciate the university and all that you guys have done for the community. It's I don't know how we would be able to manage without the support we've gotten. And I really liked Beth's comments on hope, that I think rolling out the vaccine has just been incredibly hopeful for the medical community. And we're starting to have some therapeutics, and it really is keeping everybody continuing to going so that we can get kids back to school and get back to some things we like. And have healthier, not have our hospitals full and have so much tragedy. So I really appreciate the work with UC Santa Cruz and the message of hope, because I think we do have a lot of hope right now.
Thank you. That was beautifully said. And thank you to everybody for attending. We did have some questions around whether this is being recorded and whether the recording will be provided. The answer is yes. The events are recorded. We will distribute it. There were also some questions about references that were made during the presentation by the panelists. And so I will work with the panelists afterwards to get references for those presentations and with Doug to send them out to everybody. Thank you very much for attending. Thank you to our panelists, and we are very grateful for your commitment.
I want to thank everybody who attended tonight, and please help out as much as you can, even if it's just wearing a mask outside. And remember, vaccines won't cure COVID, vaccinations will. So do your best to get out there and get vaccinated as soon as you can. Thank you very much. And this ends our event tonight.
Thanks, everybody. Thanks, Doug. Thank you to Santa Cruz Works.